Hello, my name is Sheldon Mei, and today I will be presenting my research project on identifying reactive cysteine residues within kinase catalytic sites in silico. This project was under the supervision of Dr. Matthew Shapira of the Structural Genomics Consortium and the University of Toronto. Kinases play important roles in regulating a wide range of cellular signaling pathways by phosphorylating various protein targets. This makes kinase inhibitors an interesting class of drugs that can potentially treat many diseases, including several types of cancers. Covalent inhibitors can interfere with kinase activities by forming adducts that occupy kinase catalytic sites and exclude substrates. Developing strategies of inhibiting select kinase activities to disrupt some signaling pathways and induce therapeutic effects is an important and ongoing process in the field of pharmacology. Covalent inhibitors act differently from suicide inhibitors, which also form covalent adducts with proteins. Specifically, suicide inhibitors mimic the endogenous substrate of target enzymes and need to be catalytically activated by the target enzyme to form adducts. For a covalent inhibitor to form an adduct with a protein, a reactive functional group or warhead on the inhibitor must come in close proximity to a target residue on a protein. A spontaneous reaction between the warhead and the target residue then forms a covalent bond to connect the two molecules. In the case with cysteine residues, inhibitor drugs can become linked to a kinase via a thioester bond. Cysteine residues are of interest because they are nucleophilic residues with relatively low abundance within proteins. This allows cysteines to be more selective targets for reactive inhibitor binding. Covalent binding between a protein and inhibitor molecule can be either reversible or irreversible. This depends on the chemical properties of the warhead and the target residue. Reversible inhibitors dissociate from their protein targets faster than the turnover rate of the protein. Covalent inhibitors with stronger electrophilic warheads typically form irreversible adducts which do not dissociate from their protein targets. Generally, reversible inhibitors require a sustained concentration in the blood plasma to continuously mediate their therapeutic effects, whereas the therapeutic effects of irreversible inhibitors can be induced for extended periods of time because recovery of protein activities require new synthesis of the protein target. Although cysteine is a common target residue, other nucleophilic residues like serine, threonine, and tyrosine can also be targeted. Drugs like aspirin and penicillin target serine residues in prostaglandin synthase and DD transpeptidase, respectively. There are several drugs being researched that target cysteine residues on kinases, including EGFR, ERBB2, HER2, and BTK. Many irreversible inhibitors that bind to cysteine residues incorporate Michael addition reagents like alpha-beta unsaturated carbonyl and acrylamide functional groups in their warheads. For example, dacomitinib binds cysteine 797 on EGFR to treat non-small cell lung cancer, and ibrutinib binds cysteine 481 on BTK to treat lymphoma. Researchers like Tonto et al. Found that substituting electron-withdrawing functional groups like a cyanomoiety onto the alpha carbon of acrylamides make inhibitors effective at reversibly binding to cysteines. Covalent inhibitors offer several therapeutic advantages. The high potency of covalent inhibitors allow them to completely inhibit enzymatic activities. This also means lower doses are required to achieve their intended therapeutic effects. Because irreversible or long-lasting covalent binding allows for an extended duration of therapeutic effect, covalent inhibitor drugs may be taken less often. Covalent inhibitors are also capable of targeting a range of proteins with shallower binding pockets, unlike traditional inhibitors, which require pockets with sufficient volume. High potency, low dose requirements, and extended durations of action can increase patient adherence to their medication routines. Also, protein targets are less likely to develop drug resistance to covalent inhibitors. The threonine 790 to methionine mutation of EGFR can develop resistance to traditional competitive EGFR inhibitors like gefitinib, erlotinib, and lapatinib. However, newer inhibitors like afatinib and neratinib can form a covalent adduct on cysteine 797 on EGFR to occupy the ATP binding pocket and mitigate such drug resistance. Using covalent inhibitors that target specific cysteine residues may be more efficient than constantly designing new drugs that catch up to acquired mutations. There are several important limitations to covalent inhibitor drugs that should be considered, mostly related to drug design and potential toxicity issues. Designing covalent inhibitors requires advanced bioinformatics and proteomic tools to perform computational analyses on protein structures. Such computational tools have only been around since the early 2000s. Most importantly, the hesitancy to develop covalent inhibitors has mainly been due to considerations of potential toxicity of such drugs. Drug metabolizing cytochrome P450 enzymes may act on covalent inhibitor drugs and generate electrophilic intermediates. Reactive drug metabolites can form adducts on cellular proteins and disrupt their natural functions and cause cellular toxicity. Drug protein adducts can also be immunogenic and cause idiosyncratic reactions in patients with immunodeficiencies. Drugs with reactive functional groups tend to have poor pharmacokinetic parameters because they experience high clearance rates. The therapeutic effect of covalent inhibitor drugs may be less effective against proteins with higher turnover rates, as the inhibitory effect will be counteracted by the synthesis of new target proteins. However, it is important to consider that not all drugs that get metabolized become toxic, and not all covalent adducts cause toxicity. An ideal covalent inhibitor would be weakly electrophilic and bind to selective targets. This means that they would not bind irreversibly or indiscriminately to off-target proteins like highly electrophilic warheads. This suggests that reversible inhibitors may be less toxic because off-target adducts would be able to detach more easily. Because of the relevance of cysteines towards safety and efficacy of covalent inhibitors, it is important to identify which cysteines can be accessed by inhibitor drugs and can potentially form covalent bonds with them. This study uses computational tools to assess the druggability of identified cysteine residues that line the catalytic site of defined 3D kinase structures. The aims of this project are to identify the landscape of cysteine residues near the catalytic site of kinases and determine their reactivity. This helps to predict the likelihood of reactive inhibitor drugs forming adducts on the identified cysteines. Defining the landscape of reactive cysteines capable of reversible binding to covalent inhibitors across many different kinases will help researchers design treatments that can selectively and effectively inhibit kinase activities and treat various diseases. It is of interest to compare the presence of conserved and non-conserved cysteine residues across different kinases. This helps promote inhibition of intended protein targets while minimizing off-target adverse effects. It is hypothesized that some kinases within shared kinase groups or families will have conserved cysteines. These conserved cysteines would be found at similar locations on the standard kinase catalytic domain. For example, it is expected that several cysteine residues identified in the study will be located immediately upstream of the DFG motif and near the ATP binding pocket. A bioinformatics analysis from 2006 identified 46 potential kinases with a conserved cysteine residue by the DFG motif. From the panel of 124 kinases, 18 out of 19 identified kinases with the conserved cysteine underwent Michael addition reactions and formed irreversible covalent adducts with hypothymycin. The kinase genes that were investigated during this study have been provided by the Structural Genomics Consortium Ligandable Human Proteome resource. This resource also provided sets of associated PDB codes for the catalytic domains of 267 kinase genes. Each kinase from the resource belonged to one of 8 main kinase groups: AGC, CAMK, CK1, CMGC, STE, TKL, and the other group. The AGC group contains kinases related to protein kinase A, protein kinase G, and protein kinase C. The CAMK group contains calcium calmodulin dependent kinases which are involved in calcium dependent signaling. The CK1 group contains kinases that relate to casein kinase 1, which regulate transcription and cytoskeleton functions. Kinases in the CMGC group are related to cyclin-dependent kinases, mitogen activated protein kinases, glycogen synthase kinases, and CDK-like kinases. The STE group contains kinases related to MAP2K, MAP3K, and MAP4K. The TK and TKL group contain tyrosine kinases and kinases that are tyrosine kinase-like, respectively. The kinases in the other group do not have enough sequence similarity to the kinases of the previously mentioned groups. A Python program was written to compile lists of PDB codes that produce human proteins within 3 angstroms resolution. Each list contained kinase genes belonging to one kinase group and included information such as the gene name, resolution, UniProt ID, and source organism for each PDB code. Information including the kinase group name, gene name, UniProt ID, and list of associated PDB codes were inputted to the Python program as command line arguments. The program stored the data for each list in CSV files. After the PDB codes had been generated, the 3D structures of the kinase catalytic domains needed to be generated and stored so that they could be analyzed. The software ICM Pro by Mulsoft LLC was the main tool used in this investigation to generate 3D kinase structures and perform several computational analyses on them. First, ICM Pro read the data in the CSV files and stored it in tables. For each row of the table, ICM would read the PDB code and download the corresponding protein X-ray structure from the main PDB website. ICM scripts were written to isolate protein structures of interest. The X-ray objects were converted to ICM objects so that they could be analyzed by the software. The ICM objects were saved to cloud storage. After the ICM objects have been generated, the cysteine residues lining their catalytic pockets can be identified and recorded. But first, reference kinase structures for each kinase group need to be selected. The catalytic pockets of the reference structures also need to be identified. ICM Pro could generate predicted ligand binding pockets on the ICM objects. Binding pockets that lie between the N-terminal and C-terminal lobes of the reference structure were determined to be the catalytic pockets. Because kinases have a shared structural architecture, superimposing 2 kinase catalytic domains on top of each other would allow their catalytic sites to overlap as well. ICM scripts were written to iterate through each ICM object in the kinase group, generate its binding pockets, and superimpose it on top of the corresponding reference structure. Any predicted binding pockets on the ICM object that were within 1 angstrom of the reference catalytic pocket were determined to be the catalytic pocket of the object. ICM could then identify and record any cysteine residues that lined the catalytic pockets. Cysteines that were within 3 angstroms of the identified catalytic pockets were recorded in this study. This slide depicts the reference structures that will be used for this study. These are the reference structures for all the 8 main kinase groups, and on the slide you can see that beside each kinase group there is a gene name and a PDB code. To determine the likelihood that the identified cysteine residues would covalently bind with reactive inhibitors, their reactivity status had to be calculated. The ICM cis-reactivity function was used to perform this analysis. The cysteine reactivity function uses machine learning taught by 34 reactive cysteines, 184 non-reactive cysteines, and a set of PDB proteins with covalently modified cysteines, which include 272 reactive cysteines. The function weighs several factors, including distance, between a cysteine and nearby binding pockets and the energetics of other nearby reactive residues. After the reactivity of the cysteines has been determined, ICM-Pro can arrange the protein sequences of the kinase genes in a multiple sequence alignment, and the reactive cysteines can be highlighted on their respective sequences. This helps visualize which cysteines are conserved and non-conserved. The 3D structures in this study mostly follow the typical structure of kinase catalytic domains. This image shows the structure of the human AKT1 gene with a ligand bound in its catalytic site as an example. Kinase catalytic domains have an N-terminal lobe and a C-terminal lobe with a catalytic cleft in between. The catalytic cleft binds ATP and other ligands. The two lobes are connected by a short peptide linker or hinge region. The N-terminal lobe mostly consists of a 5-stranded beta sheet and an alpha helix. The loop between the first and second beta strands is called the phosphate binding loop, or P-loop. It is rich in glycine residues, which help to stabilize interactions with an ATP phosphate. The alpha helix in the N-terminal lobe is called the C-helix. The C-helix is approximately 10 to 15 residues long and between the third and fourth beta strands. There is a glutamate residue at the beginning of the C-helix, which forms a salt bridge with a lysine residue on the third beta strand. The lysine residue also helps stabilize interactions with ATP phosphates. The first residue of the hinge region is called the gatekeeper residue. The gatekeeper residue can either allow or block access to the hydrophobic catalytic site depending on the size of the residue. The C-terminal lobe is mostly alpha helical. There are two nearby alpha helices, which will be referred to as the D helix and the E helix, that come after the hinge. The D helix is a short helix that comes immediately after the hinge, and the E helix is a longer helix that follows the D helix. There are also two short beta strands in the C-terminal lobe, which will be referred to as beta 6 and beta 7 in sequential order. There is a DFG motif and an APE motif in the C-terminal lobe. The segment in between the two motifs is called the activation loop. The conformation of the DFG motif helps regulate kinase activity for some kinase genes. I will now present the discovered cysteine locations for each kinase group. Please feel free to pause the video to look at the gene names and PDB codes if needed. When ready, just resume the video. The following slides will outline common places where reactive cysteines were located. These slides will include snapshots of the multiple protein sequence alignment of the kinase genes within each kinase group. The 3D structures of the reference kinases will be used to depict the regions where reactive cysteines were found. The regions in red represent reactive cysteine locations. The C helix, D helix, and E helix are colored orange, green, and blue respectively to help keep track of the structure orientation. The ligandable human proteome resource included 28 kinase genes in the ACG group and 237 associated PDB codes. Of the 237 PDB codes, ICM-Pro was able to generate 236 ICM objects. 77 cysteine residues have been identified to lie within 3 angstroms of the catalytic pocket of these 3D structures. 66 of the identified cysteine residues have been predicted to be reactive by ICM-Pro. There are 3 genes with cysteines 3 residues past the tip of the P-loop. These residues belong to the same kinase family. The genes RPS6Ka3 and RPS6Ka5 produce 2 catalytic domains. That is why these 2 genes appear twice in the multi-sequence alignment. The cysteines found at this location are exclusive to one of these catalytic domains, but not the other. There are nearby groups of cysteines, but their positions on the beta strand suggest that they would not face the same direction as the 3 identified cysteines in this grouping. There are 6 genes with cysteines near the beginning of the C helix. Genes from 2 kinase families share cysteines at this location. There is 1 gene with a cysteine located within the hinge region. This cysteine is not conserved at the location where it is found, and there are no nearby cysteines on the other genes in this kinase group. There are 2 genes with cysteine residues located 1 residue upstream of the DFG motif, and 5 genes with cysteine residues located 2 residues downstream of the DFG motif. While no other genes in this kinase group have cysteines located at 1 residue upstream of the DFG motif, 11 other genes have cysteines located 2 residues downstream of the DFG motif. There are 8 genes with identified reactive cysteines located within the activation loop. However, 10 other genes also have cysteines located within the same region. One cysteine residue was found within a structural domain that preceded the N-terminal lobe. The domain was in proximity to the catalytic cleft. There is another cysteine residue that was found within the N-terminal lobe beta sheet, but the sulfonyl group was facing away from the catalytic cleft. 3 genes have reactive cysteines in long tail sequences at the end of the C-terminal lobe. These sequences do not form secondary structures, but were positioned close to the catalytic pocket of their respective kinases. The CAMK group contained 37 genes. From the 483 associated PDB codes, ICM-Pro generated 478 ICM objects. 66 of the 221 identified cysteine residues have been predicted to be reactive. One gene was found to have a non-conserved cysteine residue located at the tip of the P loop. There are 5 genes with cysteines within the C helix. One gene has a non-conserved cysteine at the beginning of the C helix, while the other 4 genes have cysteines near the end of the C helix. There is another gene with a cysteine at the end of the C helix, but it was not detected by the algorithms used in the methods. It belongs to the same kinase family along with 3 other genes with conserved cysteines at the end of the C helix. One gene has a non-conserved cysteine residue in the back pocket located behind the gatekeeper residue. There are 6 genes with cysteines within the hinge region. One non-conserved cysteine was found 1 residue upstream of the 6th beta strand in the C-terminal lobe. There was only 1 gene in this group with an identified cysteine located 1 residue upstream of the DFG motif. However, another gene within the same kinase family also has a similarly located cysteine that was not identified by the method algorithms. There was one kinase structure that was being auto-inhibited by a segment of its C-terminal domain. This is a conformational state that genes in this kinase group can adopt to regulate their kinase activities. The CK1 group contained 10 genes. There were 66 associated PDB codes, and all were successfully converted into ICM objects. 15 cysteine residues have been identified, and 14 were predicted to be reactive. 3 genes have reactive cysteines within the P-loop region. 2 are located at the end of the first beta sheet, and 1 is at the beginning of the second beta sheet. One reactive cysteine is found in a back pocket behind the gatekeeper residue. The CMG group included 38 genes and 1,232 associated PDB codes. ICM-Pro generated 1,217 ICM objects. 253 cysteine residues have been identified, and 244 of them were predicted to be reactive. There is 1 gene with an identified reactive cysteine within the back pocket behind the gatekeeper residue. The location of this cysteine is shared with another group of conserved cysteines from another kinase family. The conserved cysteines from the other family have not been identified by the method algorithms. There are 3 genes with reactive cysteines within the hinge region. The 3 genes belong to 2 closely related kinase families, and the cysteines are not conserved at that location. 2 genes in this group have identified reactive cysteines near the end of the E helix. However, 8 other genes also have conserved cysteines in the same region. There is one gene with a non-conserved cysteine located one residue past the sixth beta strand in the C-terminal lobe. There are 13 genes that have identified reactive cysteines surrounding the DFG motif. 8 genes have cysteines, one residue before the DFG motif, one gene has a cysteine, 2 residues past the motif, and 4 residues have cysteines, 3 residues past the motif. One gene has a non-conserved cysteine within the activation loop. This group also contains 4 genes with reactive cysteines located within some non-secondary structures in the C-terminal lobe. 3 of these cysteines line the catalytic cleft and are conserved within one kinase family. The other lies in a region in the tail end of the C-terminal lobe that comes close to the catalytic pocket. There are 39 genes in the other group. All 393 associated P2B codes were converted into ICM objects. 301 of the 322 identified cysteine residues have been predicted to be reactive. 4 genes have reactive cysteines in the P-loop region located at the beginning of the second beta strand. There are 3 genes with cysteines located near the end of the third beta strand. This area is above the ATP binding site. 3 genes have reactive cysteines within the C helix region. 2 cysteines are found in the middle of the C helix, and 1 cysteine is found one residue before the C helix. The genes with the cysteines in the middle of the helix belong to the same kinase family. There are 21 genes with reactive cysteines within the hinge region. 2 genes have cysteines at the end of the E helix. The cysteine of one gene is conserved in related genes of the same kinase family. There are 4 genes with cysteines that surround the DFG motif. 3 genes have identified reactive cysteines located one residue upstream of the motif. There is also another gene with a cysteine at this location that was not identified by the algorithms. One gene has identified cysteine located 2 residues downstream of the DFG motif. The following genes are excluded from the multi-sequence alignment because they lack a sequence similarity with the rest of the kinases in this group. The STE group contains 27 genes. 250 of the 251 associated P2B codes were converted into ICM objects. 149 of the 172 identified cysteine residues were predicted to be reactive. There is one gene with a non-conserved cysteine at the tip of the P-loop. There are 3 genes that have cysteines, one residue downstream of the C helix. There are also several other cysteines on the multiple sequence alignment close to these reactive cysteines that were not identified by the algorithm. 8 genes have cysteines within the hinge region. These 8 genes are all members of the same kinase family. The conserved cysteine in one closely related gene did not get identified by the algorithms. There is one gene that has a cysteine residue immediately before the D helix. There are 6 genes with cysteines at one residue before the DFG motif. One gene has an identified reactive cysteine in the activation loop. However, there are 2 other genes that have cysteine residues in the same region. There is one gene with a cysteine located between the third beta strand and the C helix. Also, there is one gene with a reactive cysteine located between the C helix and the fourth beta strand. The TK group includes 61 genes. Of the 1176 associated P2B codes, ICM Pro generated 1171 ICM objects. 581 of the 646 identified cysteine residues were predicted to be reactive. There is a wide range of cysteine locations in this group. There are 4 genes with cysteines located near the tip of the P-loop. There is a conserved cysteine that is shared amongst 3 related genes within one kinase family. There is also a non-conserved cysteine located at the end of the first strand of the P loop. There are 3 genes that have reactive cysteines located within the C helix. One gene has a cysteine located in the middle of the helix, and 2 genes have cysteines near the end of the helix. It appears that these residues are mostly non-conserved where they are found. There is one gene with a cysteine located in the back pocket behind the gatekeeper residue. This residue appears to be not conserved at the position where it is found. There are 10 genes with cysteines located within the hinge region. 3 of these genes have consecutively placed cysteine residues. Cysteines in the hinge region are found in 6 different kinase families. There is a conserved cysteine residue that is shared between 2 kinase families. There are 10 genes with reactive cysteine residues surrounding the D helix. 7 of these genes have a cysteine 1 residue upstream of the D helix. 2 genes have a cysteine 2 residues upstream of the helix, and 1 gene has a cysteine 1 residue downstream of the helix. The 2 cysteines located 2 residues upstream of the helix are not conserved in other kinases of the same kinase family. Every other residue at that position is a glycine. The 7 cysteines located 1 residue upstream of the helix are found in 3 different kinase families. The cysteines are conserved in 2 families. There is another related gene with a cysteine residue located 1 residue downstream of the helix, but it was not identified by the algorithm. There are 7 genes with reactive cysteines following the E helix. 6 genes have a cysteine located 2 residues past the E helix, and 1 gene appears to have a cysteine located 3 residues past the helix. The cysteine located at this position appears to be conserved among 3 kinase families. There are 5 genes with cysteine residues around the beginning of the 6th beta strand in the C-terminal lobe. 3 genes have a cysteine located 3 residues before this beta strand, and 2 genes have a cysteine located at the beginning of the strand. The cysteines located 3 residues before the strand are conserved within one kinase family only. However, the cysteine located at the beginning of the strand are conserved across several kinase families. There are 5 genes with reactive cysteines 1 residue before the DFG motif. It appears that this cysteine location is only conserved between 2 kinase families. There are 22 genes in the TKL group. 201 of the 203 PDB codes have been converted into ICM objects. 55 cysteine residues have been identified, and 49 were predicted to be reactive. There is 1 gene with a reactive cysteine located at the end of the first beta strand within the P loop region. 3 genes have reactive cysteines within the hinge region. These cysteines are not conserved within their respective kinase families. There are 3 genes with cysteines located 1 residue before the DFG motif. These cysteines are not conserved, but 2 are found in genes belonging to the same kinase family. Although the software was able to predict 1 cysteine on IRAK4 was reactive, the 3D kinase structure appeared to be disordered in the N-terminal lobe. The beta strands did not appear to accurately represent the typical kinase N-terminal lobe, and the detected catalytic pocket was shaped inconsistently compared to other PDB codes for the same gene. This study managed to define the landscape of reactive cysteine residues lining the catalytic cleft of various kinases. The table and stacked bar graph depict the different reactive cysteine profiles amongst the different kinase groups. The graph and table show that there are several hotspot reactive cysteine locations. Some of the most common reactive cysteine locations includes the hinge region and areas surrounding the DFG motif. The data also suggests that there are several potentially viable cysteine targets in the P-loop region and the C helix. A recent review by Knapp and colleagues outlined several cysteine locations on kinase active sites that can potentially bind covalently with inhibitor molecules. Many of the identified cysteines from this study match cysteine locations reported in the review. This includes cysteines on kinases with known crystal structures and known covalent inhibitor targets. However, this study also found several reactive cysteine locations that were not explicitly outlined in the review. This includes areas around the C helix, the end of the E helix, and regions around the two beta strands in the C terminal lobe. This suggests that the methodology of this study was effective at predicting reactive cysteine residues, including ones that have not been studied in depth. However, more research is still needed to see if these locations can be successfully accessed. Looking at multiple PDB codes for each gene allowed the algorithms to consider a wide range of possible kinase conformations and catalytic pockets. Repetitively identifying the same cysteine residues from multiple 3D structures provide confidence that the identified cysteine residues were viable drug targets instead of being identified by chance. However, this was not possible for some genes that only have one or only a few PDB codes to inspect. Perhaps some conserved cysteines that were not successfully identified in this study may be identified if more structures were inspected. While the algorithms in this study were mostly reliable to predict which cysteines were viable covalent binding targets, it is important to manually confirm that the identified cysteines can actually be accessed from the catalytic cleft. For example, in this study, cysteine 221 on GRK2 was excluded from consideration as a viable cysteine target that can be accessed by the catalytic cleft because it was facing the wrong direction on the beta sheet. The algorithm also works best when it is analyzing well-defined proteins that follow the traditional kinase catalytic domain structure. Some 3D structures with improperly folded kinases cannot be relied upon to produce accurate reactive cysteine identifications. Improperly folded kinases that produce abnormally shaped catalytic pockets may be near a reactive cysteine by coincidence. This was seen with the IRAK4 gene in this study. The one misfolded protein produced a reactive cysteine result, but no other proteins of the same gene produced any reactive cysteines. While the algorithm was able to detect several conserved cysteine groups, there were still some that were not captured in the results. Perhaps the algorithms can be adjusted to identify cysteines that are slightly beyond the 3 angstrom range from the catalytic pocket. This may allow some conserved cysteines that were not captured in the results during this investigation to become successfully identified. Overall, the results were able to identify both conserved and non-conserved cysteine residues. Conserved cysteine residues within families of kinases may not be ideal targets for covalent inhibition. An inhibitor may covalently bind and inhibit the activity of closely related kinases. This may cause off-target adverse effects or toxicity. There are several unique non-conserved cysteine residues within each kinase group. These non-conserved cysteines that are isolated away from conserved groups of cysteines would be better targets to facilitate more selective kinase inhibition. This study was successful at mapping the landscape of reactive cysteines across the main kinase groups. The results of the study matched several previously discovered cysteine locations and drug targets reported in literature. This suggests that the methodology and algorithms used in this study can accurately predict viable covalent inhibitor binding sites. There are several directions future investigative steps can take. More research is still needed to confirm if the identified cysteines from this study would make viable covalent drug targets. It may be beneficial to investigate if considering binding pocket hydrophobicity and electrostatic profiles allows for more selective covalent inhibition. Future investigative steps may also be to use similar procedures and algorithms to expand the analysis onto larger fields of human proteins. This may include defined protein crystal structures and predicted protein structures from resources like the AlphaFold Protein Structure Database. Continued mapping of the human cystenome will help researchers design treatments that can effectively and selectively inhibit protein activities and cellular functions to treat many diseases. Thank you for watching my presentation. Have a nice day.